Hello students, welcome to Trunkie Academy. We are studying about the biotechnology, and in the last class yesterday we studied about the tools of uh, recombinant DNA technology. Okay, and before that we studied about some important terminologies and some important basic concepts about how there are different principles that's involved in the biotechnology, principle and processes. Okay, so now today we will study about the biotechnological applications in agriculture. Since the time these biotechnological tools are developed, these are widely used in medicine, agriculture, and some other field for the welfare of human beings. Okay, so in introduction we see that biotechnology essentially deals with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, okay, or microorganisms, fungi, plants, and animals. Okay, so it essentially deals with the industrial scale production at the large scale for the supply to common people for the use as the biopharmaceuticals or biologicals or the using genetically modified microorganisms, okay, or fungi or plants and animals. Okay, so this is about the application, brief introduction. The application of biotechnology include therapeutics, medicines, okay, diagnosing the different diseases with the help of biotechnology, okay, and genetically modified crops for agriculture. We know that certain crops are producing the good quality grains or fruits, so that we need it, and there are some type of the plant crops that are not producing the fruits and good quality crops. Okay, so what we do in the with the help of the biotechnology is that we genetically modify that particular crops to get the desirable trait or desirable character, so that we will get the good quality fruits or seeds or grains. So that will be for the useful for our, that will be for our interest and for the use for the benefits of the human beings. Okay, now that is the genetically modified crops for agriculture. Okay, processed food. Okay, food are also processed with the help of the biotechnology. There are so many nowadays. We see that the packed foods are available in the market, and these are also the processed food, and so these should not be decomposed, should not deteriorate in its quality, to ensure that some biological techniques are applied, okay, and it is sold in the market. Bioremediation is another important application of the biotechnology. Okay, we know that there are so many different kinds of water pollutants, sewage, and some other kinds of water pollution that may be very harmful for the aquatic bodies. So these bioremediation is actually changing that sewage or any water pollutant into different forms. We are also applying this biotechnological process to change the sewage or any other pollutant or substance to into some useful products or useful substances. Okay, like injection recovery, you can see the carbon dioxide, oxygen, and nutrients. Okay, the contaminants that are there, and with the help of the bioremediation, it is the quality is improved. Waste treatment, different kinds of wastes are produced in. We know that there are hospital waste, medical waste, domestic waste, industrial waste. Okay, there's so many different types of waste, are urban waste. Okay, so all these wastes are treated with the help of biotechnology, and there is energy production also with the help of the biotechnology, energy production. Okay, now you can see the farm, the value of the usage, the forestry, highway, office. That means we highly efficient biogas production. So biogas production is one of the application of the biotechnology. The low cost waste treatment, electricity cell, and that is the one of the application of the biotechnology. Okay. Three critical areas of biotechnology are providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. One of the most important, okay, you can say out of the three critical areas, okay, three critical areas or three critical important application, okay, or use of the bioenergy is the one is the providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme company, and the creating optimal conditions through engineering or a catalyst to act, and downstream processing technologies to purify the protein organic compound. So these are the critical area of biotechnology, okay, where these are done. Okay. Biotechnology applications in agriculture is that three options that can be used for increasing food productions. Three options are there, okay, for increasing them with the help of the biotechnology. Agrochemical based agriculture. Agrochemical means the it is a kind of chemical which is the used for the increase the production in agriculture and agrochemical. Okay, so organic agriculture where it is completely organic, okay, no chemical or artificial fertilizers are used, it's called organic farming. And genetically engineered crops based agriculture, that means some of the crops are genetically engineered in such a way that they will produce good quality crops, okay, as desired by us, okay. So that is the now around 1960s, several countries, including India, experienced substantial and dramatic increase in agricultural production, which was termed as Green Revolution. Okay, around 1960s. Okay, so there was a huge sudden increase in the production of the agricultural crops, and that was experienced by several countries, including India also. So that was termed as Green Revolution by William Gould. Okay, USAID. This is the organization to which he belongs. Okay, William Gould. So United States Agency, International Development, that is the organization to which he and he gave the term, okay, Green Revolution, because this Green Revolution brought the a large quantity of production of large quantity of the agricultural crops. Okay. Now Norman Borlaug, who initiated the efforts that led to Green Revolution. Because he is the main person who was involved in the this Green Revolution. Okay, so his efforts were very substantial and very much appreciated, and that is the reason he is regarded as the father of Green Revolution. Because of him only there was, okay. Dr. M.S. Swaminathan and his team were instrumental for success of Green Revolution in India. So Dr. M.S. Swaminathan, he is an Indian scientist, and he was one of the very important person, and he was very much instrumental, played a very critical role in the success of the Green Revolution in India. Okay, so Dr. Norman Borlaug, he was the main person to initiate the the Green Revolution and contribute to the Green Revolution. And Dr. M.S. Swaminathan, he was the person and the scientist from India to play a very critical role in the success of Green Revolution in India. Okay, so Green Revolution was possible due to a number of factors, both economical and scientific, and include the use of improved varieties, chemical fertilizers, and land reforms, pesticides, and better agricultural management strategies. Now try to understand this. Green revolution was possible due to a number of factors. It is not that they have produced some kinds of fertilizers, okay, and some kind of injecting some kind of medicines or some kind of important hormones injecting into the crops, and then after 1 month or 1 year there was a huge production of the crops. It was not like that. It was not like that. There were many reasons for the successful success of these green revolutions, and they were dependent on improved varieties. Okay, that means we are hybridizing to obtain the good varieties. For example, I'm telling you, there is one variety which is giving a good quality mango, and there is one other plant which is not giving good quality mango, but it is disease resistant. Try to understand this. There is a crop which is not producing good quality fruits or grains, but that is the disease resistant. Another is not disease resistant, but is producing a good quality grains or fruits. So we are hybridizing, and we are obtaining such a progeny, offspring, and hybrid that will have the disease resistant capacity, as well as it will produce good quality grain or fruits. Okay, so improved varieties. Now chemical fertilizers also played an important role in the success of green revolution. Okay, um, land reforms. Okay, how the land should be used in agriculture. Okay, that is important. How it should be used, how much should be used, and how much should not be. There should not be any kind of construction and of the buildings or the structure to disturb the agriculture. Pesticides are to control the pests, that the kind of insects which feed on and damage the crops. So using the some kind of pesticides that kills the pests and allow the crop to grow better. Okay, so better agricultural management strategies, different kind of strategies for irrigation and other management strategy were important and very much played a vital role in there. Okay, so another is improved irrigational facilities, because the growth and the good yield of the agricultural crops depend on the improved, or you can say the good irrigational facilities. Okay, so these were the different reasons, even the factors that led to the success of the green revolutions. Okay, so even though green revolution succeeded in tripling the food supply, it increased the crop by 3 times. It was not enough to feed the growing human populations. Though it increased 3 times, okay, but the human population was huge, and it's still very huge, okay, and this was not sufficient to supply. Now, another is the green revolution. Increased yield have been partly been due to the use of improved varieties, but mainly due to the use of better management practices, and also use of agrochemical fertilizers and pesticides. Okay, so that better management practice, how to manage it, that how to grow the crops, how to grow different crops in the same field. There are some crops, okay, they are intercropping, mixed cropping, all these things. Okay, so better management practice and the use of agrochemical fertilizers and pesticides. Okay, however, for farmers in the developing world, agrochemicals are often too expensive. That will be the, and they are normally avoided because of the cost, and they show harmful effects on the environment also. Agrochemicals, so they have got some harmful effects on the environment. They cause some pollution and harmful effects on the environment. That means the harmful effects on both plants and animals, including human beings. Okay, so using of pesticides like DDT is very harmful. When it was discovered, it was used by farmers indiscriminately, and they were not knowing the consequence of this use of this DDT, and that led to the extinction of some swift flying birds. Okay, so someday when I will teach while teaching about the ecosystem ecology, I will explain about, when we will study the topic from the environmental issues, I will definitely explain about the how this use of DDT led to the extinction of some swift flying birds. Okay, for the increase in yield with existing varieties are not possible using conventional breeding. Yes, it's not only, simply there is one variety which has got good grains and not disease resistant, and one variety which has disease resistant but not good grain, but not good resistant. So we'll just cross, and new offspring will have good grains and disease resistance as well. It's not so easy. Okay, it's not only that. There are some biotechnological process we need to apply to get the good quality and good breed. Okay, so use of genetically modified crops is a possible solution. Genetically modified, we interfere, we change the genome of the plant, genome that the crop, to so as to obtain the good quality crop. Okay, so wild relative and the crop plant, then conventional breeding. You see? Now insect resistant line, that means the CRI1AB gene, and herbicide tolerant line, ESPS gene. So when they crossed, and then we got the insect resistant and herbicide tolerant hybrid both. So that was the genetically modified crop. In this way, with the advent of the biotechnology, especially the genetic engineering, a new ray of hope to increase the food crop and to reduce the use of chemical fertilizers and pesticide has been envisaged, which might lead to other type of revolution called green gene revolution. Okay, so gene. So with the discovery of with the advancement of biotechnology, especially the genetic engineering, a new ray of hope to increase the food production and the use of chemical fertilizer has been envisaged. Now this has given a hope that now we can produce more and more crop, okay, and we can reduce the use of chemical harmful pesticides and fertilizers that has been that is okay, and lead to the other type of revolution, gene revolution. Okay, so bacteria has got some kind of a circular DNA called plasmid. We can integrate some kind of uh, part of the gene or DNA into that plasmid, so that we get our desired trait. Okay, so that cloning and the designing genes, then transformation, tissue culture, okay, and then we got the plant of our desired trait. Okay, so that is the gene revolution. The future of agriculture is dependent on gene revolution. Now, with the help of genetic engineering, it is possible to break through the natural species barriers and systematically transfer genes from one species to another that may not be possible in nature. Naturally, that is not possible that one gene will be transferred to the another species. So that was possible with the genetic engineering. Okay, so cutting out the one gene or taking out one gene or DNA sequence from the DNA molecule and then integrating with the plasmid or the bacteria, and then getting the designing and cloning, and then inserting into the host for the desired trait. Okay, all these things. The gene revolution is likely to provide sound agricultural production. Yes, with the help of gene revolution, okay, what we can do is we can get the good quality crops by changing the gene or by changing the genome of the crop plant. Okay, because the particular, we know that if we know that if this particular gene is responsible for this quality or this responsible for disease resistant, and this gene is for a good quality good size of the fruit, this gene is responsible for a good taste of the fruit, this gene is responsible for good grains of the crop, so then we can genetically modify and we can get the, so gene revolution is likely to provide sound agricultural production. Okay, so now the substantial increase in agricultural production is termed as the white revolution, or is it the black revolution, green revolution, or it is the red revolution, it is the green revolution. Very simple questions. Okay, father of green revolution in India is Dr. M.S. Swaminathan. Norman, okay, Dr. M.S. Swaminathan. He is Indian and he has played a very critical role in the success of green revolution. Okay, so it is Dr. M.S. Swaminathan. Okay. Now we come to genetically modified or the GMO. Okay, so what are these GMO? We see genetically modified bacteria, plants, bacteria, fungi, and animals whose genes have been altered, manipulated. Okay, and altered by manipulation are called genetically modified organisms. Okay, so this means these are the different some organisms, maybe plant, bacteria, fungi, animals. Okay, so we change the genome. Okay, we change the we do some manipulation in the genes. Okay, and alter the gene to get the good variety, a good traits. Okay, that is a genetically modified organisms. Okay, GM plants or genetically modified plants are useful in many ways. Production of high yielding and disease resistance varieties we get from genetically modified plants. Okay, they are good yielding, they produce in large number. Okay, produce is good, yield is good, and also they are disease resistant. Okay, so that is the advantage. Another is genetically modifications have made crops more tolerant to abiotic stress, cold, drought, shade, and that is the reason the produce is good, and that is the advantage we get. Okay, genetic modification made the crops tolerant to abiotic stress like cold, drought, salt, and heat. So they are growing well, even in the presence of abiotic stress. Okay. Reduced reliance on chemical pesticide pest resistant crop, because there are some kind of crops which are genetically modified and they do not, because some even in the presence of the pest also they are growing well and the pests are not able to affect and the crop plant, okay, rather the crop plant are affecting the pest, okay. So that means the bt cotton is one of the very good example. Many researches and studies were done on this plant to improve the quality of this cotton, and the bt cotton is one of the example, okay. So chemical to reduce the, how it was done, that help to reduce the post harvest losses, okay. Biotechnological process and the genetic engineering and the gene revolution help to reduce the post harvest. Sometimes what I mean, after harvesting, okay, after cutting the and obtaining the harvesting of the crop plant, then there is some kind of damage. It will be damaged either by the pest or some due to some kind of due to moisture, okay, or due to some other reason the harvest is damaged, lost. So that loss was also reduced due to the biotechnological process and the genetic engineering, okay. So it remained fresh and not getting deteriorated, okay. Increased efficiency of mineral usage by plant, this prevents early exhaustion of fertility of soil. So increased efficiency meaning what they are doing, the crop plant is using the minerals efficiently, and this reduce the the exhaustion and finishing of the scarcity of the mineral from the soil, okay. Exhaustion of the fertility of the soil. So that is the another increased efficiency of the usage of the minerals, and hence the nutritional value of food, because we are growing the crop plants of our interest. So we want the nutrients to be as per our interest. That means vitamin A enriched rice, okay. So that is they provide. So that is also possible due to biotechnology and the genetic engineering, okay. So another, the genetic engineering has been used to create tailor-made plants. Means tailor-made means according to our design, according to our wish, okay. The plant that supply alternative resources to industries in the form of star fuels and the pharmaceutical, that is called the tailor-made plants, okay. The chain of being potential pathways for biofuel production, okay. The chain what they are doing, sunlight, then the biomass, okay. There is the biomass, that means the all the natural vegetations, okay. Thermal treatment, then there is a kind of syngas, okay. Microbes or catalysts, then these biomass are monomers. They are the depolymerized and they form the monomers, and then this is used as the fuels, okay. So it's called tailor-made fuels. What we are doing, we are actually using as the fuel as a tailor-made instead of the other fuel. Some of the applications of biotechnology in agriculture are production of pest resistant plant, very important, because if you are using the pest, this is harmful for the environment as well as for the ecosystem and even plant also, though it is killing the pest, but it's harmful to the plant to some extent, because it reaches down with the rainwater and it goes into the soil, which is harmful for the ecosystem and environment, which could decrease the amount of pesticide uses. So by creating and producing the pest resistant plant, we are able to reduce the use of pesticide, okay. So Bacillus thuringiensis, BT for short, okay. Bacillus thuringiensis, okay, that is the name of the bacteria, the short form is called BT, okay. So Bacillus thuringiensis, this is the name of the bacteria, and that BT gene is inserted into crop, BT gene. So what we are doing, this particular bacteria, Bacillus thuringiensis, this has got some one gene that will produce a kind of substance or protein that will act against the pest which is damaging the crop. So what we have done is we are taking the gene of that bacteria, Bacillus thuringiensis, okay, and inserting into the crop plant, so that this crop plant will also produce a kind of substance which will act against the pest, and the pest will die, and we do not need to use the pesticide. So crop is infected with European corn borer. Now when it is inserted, the pest dies when feeding on any plant. Why? Because this plant with the gene of the bacteria Bacillus thuringiensis, and that gene which is producing a type of substance against the pest. That due to that this pest is not surviving and died, okay, and could not grow on the crop plant. Bt toxin, that means what is the things that made to die about this pest, that is the Bt toxin, a kind of toxic substance is released, okay, and that causes the death of the pest, okay. So here you must understand that Bt toxin is produced by a bacterium called Bacillus thuringiensis. This is a technique how the scientists have researched and found, and then they have applied biotechnology to prevent the pest and to prevent the damage of the crop plant, okay. So what they have done, a Bt gene is transferred from Bacillus into the corn, okay, crop plant, and this is helpful in the damage, to prevent the damage by uh, pest, okay. So the Bt protein penetrates and the collapses the cells lining the gut of the insect and died. So what happens when Bt protein that goes into the, what happens, it collapses and it and the lining of the gut, and this pest die, okay. The borer, the European corn borer feeds on the corn plant and ingest the protein encoded by the Bt gene. So when these pests are feeding on that, so they are ingesting the protein encoded by the Bt gene, and this what they have ingested, the protein, that protein is going to interfere, and they penetrates and collapse the cells lining the gut, and they die. So that is how the biological way of controlling the pest. Bt toxin gene has been cloned from this bacterium and has been expressed in plants to provide resistance to insects without the need of insecticides, in effect created biopesticides. So this is the way of controlling the pesticide biologically, and hence it is called the biopesticide, because we are not applying, we are making the plant resistant to the pesticide, resistant to the pest, okay. Biotechnologically, by altering and manipulating the gene of the crop, we are making this plant resistant to pest, okay, and so that we do not need to use the pesticide. The examples are Bt cotton, Bt corn, rice, tomato, potato, and soybean, etc. These are the examples of the Bt toxin gene, in which the Bt toxin gene are used to prevent the pest infestation. Okay, so GM example, insect resistance through introduction of the Bt gene. Okay, that I told you. Now, Bt cotton. Some strains of Bacillus thuringiensis produce proteins that kill certain insects, such as the Lepidopterans. Lepidopterans are the one group of insects, okay, in one group of the arthropodans that are called the Lepidopterans: tobacco, birdworm, armyworm, okay, coleopterans, beetles, and dipterans, flies and mosquitoes. So they some strains of Bacillus thuringiensis produce such a kind of protein that kill the certain insects, such as Lepidopterans, that is the example of tobacco, birdworm, or armyworm, coleopterans, beetles, and dipterans, flies and mosquitoes. So these are the types of some insects that are easily killed by the toxin produced or toxic proteins or toxic proteins that are produced by the Bacillus thuringiensis. Okay, so cotton ball, okay, are destroyed. Okay, ball, a fully mature cotton ball. Okay, so you see A and B. Now you see Bacillus thuringiensis forms the protein crystals during a particular phase of growth. Okay, so these crystals contain a toxic insecticidal protein. Okay, so Bacillus thuringiensis produces a kind of toxic protein or toxic protein crystals during a particular growth phase. It's not that it's continuously producing. Okay, so a certain phase of this growth is producing, and actually the Bt toxin proteins exist in inactive prototoxin, but once an insect injects, okay, an inactive toxin converted into an active form or toxin due to alkaline pH of the gut of the cellulized. What happens when they are feeding on the part of the crop plant which contains the Bt toxin gene, and the Bt toxin protein is produced? So this Bt toxin, when they are ingesting, that is actually before ingesting is in the inactive form as prototoxin, not active form. But as soon as it enters in the gut of the insects due to alkaline pH. Okay, it is converted into the active form, and that's alkalinity, the alkaline pH solubilize the crystals and make these crystals and convert into active toxin. Okay, from prototoxin to active toxin, protoxin to. So these are Bacillus thuringiensis. Okay, beta-toxin crystal. Okay, crystals are ingested. Okay, the solubilization of the crystal by alkaline pH, then activation of the toxin, and then the killing and the destroying the pest. Okay, so this is how you can see the picture is given here. Insect mid-gut cells. Okay, the mid-gut cells. Okay, mid-gut means the interior gut, mid-gut, the center. So there is a gut. Okay, from mouth to anus. The central in the middle, that's called mid-gut. Okay, so in the mid-gut you can say the binding to the receptor, toxin monomer, and they are toxin the oligomer. Okay, and then the GPI and cut proteins are there, and membrane insertion, and then the cell death. Okay, pores lead to osmotic lysis. Okay, and then there is the, so that's how activation of this protoxin causes the death of the pest. The activated toxin binds to surface of the midgut epithelial cells and creates pores that cause the cell swelling and lysis, and eventually they cause the death of the insect. What is doing actually, when they are ingesting that crystals form, okay, which is the protoxin, the alkaline pH is solubilizing and converting into toxin, and these toxins are binding to the cells or that present in the midgut epithelial cells, the midgut, and that creates the pores and causes the swelling and lysis and breaking and the tearing of the cells, and eventually cause the death of the insects. Okay, specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. There are a number of them. For example, the proteins encoded by the genes CRY1AC, CRY2AB control the cotton bollworms, while that of CRY1B control the corn borer. So different genes are used. So the toxin is coded by a gene named CRY. Okay, so the choice of genes depends upon the crop and the targeted pest, as the most beta toxin are insect group specific. Okay, so it depends on the which particular gene we will insert into the crop plant. That depends on the crop, and which pest is affecting the crop. It all depends on the type of the pest that is targeted and the crop plant on which we are inserting. Okay, so pest resistance plants are nematode resistance plant. Several nematode parasitize a wide variety of plants and animals, including human beings. Nematode Melodigine incognitia infects the root of the tobacco plants and cause a great reduction in yield. So root knot, you can see this. Yeah? So Melodigine incognitia. Okay. RNA interference, interfering with the RNA. RNAi in short, it takes the RNA interference. So in short we say RNAi in short, but takes place in all eukaryotic organisms as a method of cellular defense. Okay, so that means defense response that is mediated by the cell. This method involves the silencing of a specific mRNA due to a complementary RNA molecule that binds to and prevents translation of mRNA. Okay, that is silencing. So what we do, we are making the stopping the expression of one particular gene. Okay, by silencing the specific mRNA. So that mRNA, although it will be DNA will encode the mRNA for the production of the protein or amino acid, but before the expression, before this translation of the mRNA as directed by the DNA, we stop. We are not allowing that mRNA to express to translate into the amino acid. This mRNA are being silenced due to a complementary RNA molecule that binds to and prevents the translation of the mRNA. That is called the mRNA silencing. We are not allowing that mRNA to translate and form an amino acid. Okay, so that is called the mRNA silencing. The source of this complementary RNA could be form an infection by virus having RNA genomes or mobile genetic elements, transposomes that replicate via an RNA intermediate. So this is the of how we are going to silencing the even bacterial composite transposome, okay, or then we have the transposable elements, okay. So these are the now viruses having RNA genomes, okay. So that the one we are going to complementary binding, so that the mRNA will not be able to translate, okay. So RNA interference mechanism using agrobacterium vector, nematode specific genes were introduced into the host plant. Using this technique, RNA using agrobacterium vectors, nematode specific gene, which nematode is going to affect the or parasitizing, depending on that, the genes were introduced in the host plant to prevent the effect of nematode, okay. The introduction of DNA was such that it produced both sense and antisense RNAs in the host cell. So it was produced in such a, it was applied in such a way that it produced both sense and antisense RNA in the host cell, and these two RNAs being complementary to each other formed a double-stranded RNA. And what happened? This long double-stranded RNA molecule are cleaved to produce as single RNA by dicer, okay. And dicer is also known as the endoribonuclease dicer or helicase, is an enzyme that cleaves double-stranded RNA into short double-stranded RNA fragments called small interfering RNA and microRNA respectively. So dicer is important for exam. Dicer is also known as endoribonuclease because it is cleaving the RNA molecules, or it's also called helicase. It is an enzyme that cleaves the double-stranded RNA into short double-stranded fragments and into small interfering RNAs and microRNA respectively. Okay, so that is how. Now, single RNA molecules are single-stranded RNA molecule or single single-stranded RNA interference. Okay. RNA molecule are then incorporated into the multi-protein RNA inducing silencing complex. So first we are getting this RNA molecule, then we are incorporating into the multi-protein RNA inducing silencing complex. This is called RISC, RNA inducing silencing complex. So mechanism RNA interference, what do you think? Synthetic siRNA and then the double-stranded RNA, dicer. Okay, dicer is, then we are getting the fragments, okay, siRNA, and then we are using this strand separation, okay, RISC, natural process RNAi, and complementary pairing, okay, and then again there is the cleaving, then mRNA is there, and then this is the cleaved mRNA, the mRNA degradation, therapeutic gene silencing. So this, after this cleavage, okay, cleaved mRNA is no more functional, and this is the therapeutic one. We are not allowing this DNA to express and translate, and thus we are silencing. This is called therapeutic gene silencing. Deliberately, we do not want to this to. Okay, so duplex RNA is unwounded leaving an antisense strand to RISC to complementary mRNA for subsequent endonucleolytic cleavage. Okay, so duplex that has formed is unwounded. Okay, leaving antisense strand. So there is sense strand and antisense strand to RISC, like in the sense in the diagram RISC. Okay. So complementary pairing, So then after this, we are since this is mRNA is formed. Okay, now we see the duplex RNA unwound, leaving antisense. Okay, so it is unwound. Okay, here you can say unwounding, and then after unwounding there is a cleaved mRNA which is a non-functional. The consequence was that the parasites could not survive in a transgenic host expressing specific interfering RNA. Okay, so they were not able to, the parasites or the kind of different worms, they were not able to survive because of this. Okay, because of this specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Okay. Now we'll go through some questions. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. Okay, so it is number 2. BD toxin is produced by the bacterium Bacillus thuringiensis. Okay, so it is the number 3. The mechanism which involved the silencing of mRNA due to complementary RNA is the DNA interference. It is the mechanism which involves silencing of a specific mRNA due to it is the RNA interference. Okay, so it is the RNA interference. We are interfering and we are making it silent and not allowing the its translation. Beta toxins are degraded in the gut of insect at alkaline pH. So it is not acidic, it's not it's alkaline, it's not neutral, this is the alkaline pH because that is dissolved, the crystals are dissolved, and then it becomes active. From protoxin to it becomes the active toxins, and then they are going to bind to the epithelial cells of the midgut, and that cause the pores, holes in the cells, then cause the swelling and ultimately the dying of the death of the cells and ultimately death of the that pest. Okay, so that is how bt toxin works and makes the crop plant resistant and protective from pest. Okay, so this was more about the application of biotechnology in agriculture. Okay, so how it is used, applied. Now tomorrow is Sunday, and day after tomorrow on Monday we'll study the next topic. Okay, so after this class, as I have told you already, I will be sending one DPP daily practice problem. You go through that. When you log into your academic booster, when you click on the resources, there you'll find some links on the left hand side. There you'll find the writing material. By clicking on writing material, you will get DPP. Download that, answer those questions, and send back to us for evaluations. Okay, so thank you for listening and joining today. Again, Monday we'll meet at the same time. Till then, goodbye. Take care.